Okay. In this second segment, we're going to look at a little overview. So, basically, an overview includes three major steps of cellular respiration or aerobic respiration. More specifically, this includes glycolysis, the citric acid cycle, and oxidative phosphorylation. Now, there is a step in between glycolysis and citric acid cycle, which is kind of important. And I think your textbook kind of combines it with citric acid cycle, but only because it occurs in the same space under the same conditions. So we'll kind of talk about that when we get to that point. So glycolysis is really just the splitting of glucose into two molecules. Remember, glucose is a 6-carbon molecule. It's split into two 3-carbon molecules, and that molecule is called pyruvate. Then pyruvate becomes oxidized, and that's that step that's in between there. It's called pyruvate oxidation. And then once pyruvate is oxidized, it becomes a molecule that is allowed to enter the citric acid cycle. The citric acid cycle actually completely breaks down the glucose molecules. So glycolysis is like the partial breakdown, basically the splitting of glucose. Citric acid cycle results in basically no more glucose. And then the final step is oxidative phosphorylation. This is where the majority of the ATP for the cell is made under aerobic conditions. First, we want to talk about the types of phosphorylation. Now, this particular chapter only covers 2 of the types. Now, there's a third type of phosphorylation in chapter 10, and that's related to photosynthesis. But this becomes important because the type of phosphorylation occurs at different places, only generates a certain amount of ATP. So let's talk about that. So there's oxidative phosphorylation. So like I said, that's the final step in aerobic respiration. This is where the majority of the ATP is made, specifically about 90% of the ATP. It consists of a ton of redox reactions. The rest of the steps in the cycle or in the pathway involve redox reactions as well. But again, this is the general trend here. Redox reactions are always going to be involved. And what this is oxidative phosphorylation includes 2 major phases. One of them is electron transport. So remember, I was talking about the electron transport chain. Well, this involves the electron transport in the electron transport chain and what's called chemiosmosis. We'll talk more about that when we get to that step in a separate segment. Then the other type of phosphorylation is called substrate level phosphorylation. Now, substrate level phosphorylation occurs in glycolysis and occurs in the citric acid cycle. Oxidative phosphorylation occurs in that particular step. It's oxidative phosphorylation. Now, there's a major difference between the two, and I'll kind of point this out as we go through these steps within the segments. But first, we're going to talk about oxidative phosphorylation because we're going to be dealing with glycolysis in the citric acid cycle first, and then we'll be dealing with oxidative phosphorylation since that is the final step in aerobic respiration. All right. Substrate level phosphorylation involves basically the transfer of a phosphate from a molecule, which is a substrate, okay, to ADP. Remember, adenosine diphosphate. Okay, so this is ADP. It needs another phosphate in order to become ATP. So this is how ATP is generated by substrate level phosphorylation. The phosphate comes from a substrate, and there's usually an enzyme that helps catalyze this chemical reaction. So basically, the substrate is going to be released without the phosphate, and then, of course, the phosphate gets transferred to ADP to make ATP. Now, we're going to talk about oxidative phosphorylation much later. However, I just want to mention here is that a major difference is that the phosphate is inorganic phosphate, which is combined with ADP to make ATP. Okay, so this is considered an organic phosphate because it's attached to an organic molecule, the substrate. All right, so let's move on. We can use money as an analogy to talk about the energy that's derived in this process. Okay, so in terms of glucose, glucose is like a larger denomination bill. Okay, it's worth a lot, so it's holding a lot of energy, but it's hard to spend. So what we mean is it's hard to break down, okay? Or it's hard to get that energy. The energy must be extracted from glucose. Now, ATP is just like a bunch of equal smaller denomination bills. That could be spent more easily. It's already available to do work. Cellular respiration, the whole process, allows the cell to cash in on the large denomination bills, the glucose, right? In order to make smaller denomination bills, the ATP, which can be used more readily. Okay, so with that in mind, we're going to kind of start with that, and that's glycolysis. So this image really kind of shows, it really explains a lot. Okay, so I am a firm believer in pictures because pictures can tell a lot more, just as much as text, if not more. Now, we can see a mitochondrion here, and we can see the process of glycolysis. Obviously, glycolysis does not occur in the mitochondrion. Okay, so glycolysis actually occurs in the cytosol. And what glycolysis is basically, remember, the splitting of glucose into two pyruvate molecules. During this process, electrons are extracted and then transferred to NAD plus to make NADH. So you can see NADH is made here. And during this process, ATP is made directly by substrate level phosphorylation. Remember, we just talked about that. Now, again, with that in mind, glycolysis, we're going to talk about glycolysis a lot more in detail. Okay, so just keep in mind, this always occurs in the cytosol. So what we're looking at here, this kind of gives you an overview of what we're going to be moving into. So remember, glycolysis is really the only thing that happens in the cytosol. Everything else in aerobic respiration occurs in the mitochondrion, and that's why the mitochondrion was there in that first image. Okay, so pyruvate oxidation, the citric acid cycle, oxidative phosphorylation, they all occur in the mitochondrion. The only thing that doesn't is glycolysis. So a little bit more about glycolysis. Remember, it's the splitting of sugar or glucose. Here we're going to talk about the glucose. Remember, glucose is a 6-carbon molecule. If we split it in half, we create 2 3-carbon molecules. Those 3-carbon molecules are called pyruvate. Glycolysis occurs in the cytoplasm. We already established that. Now, there's 2 major phases of glycolysis. There's what's called the energy investment phase and the energy payoff phase. So kind of continuing with that analogy of money. Okay, so remember, when you invest money, you're kind of putting aside money. You're putting money in order to make more money. Okay, so we can think of our money as ATP. So here we're going to have to use some ATP or spend some ATP in order to make more ATP. So the energy investment is the spending of ATP. The energy payoff is the making more of ATP, so the building upon it. Again, glycolysis occurs in the cytoplasm, and it occurs whether or not oxygen is present. So glycolysis can occur in the absence of oxygen. Glycolysis can occur in the presence of oxygen. Let's look at the energy investment phase. Now, you're not going to have to memorize all of these enzymes and all these structures in this particular course. If you took a more upper level course, molecular biology or biochemistry, you may need to know these structures and be able to draw them. However, the point here is to show the investment of ATP. Okay, and I'm going to point out some other things. So, I've already talked about ASE. Anytime a word ends in ASE, it's an enzyme. Often the enzymes are named based on their function. So here, this one says hexokinase. It must be, so hexo means 6. So here we're dealing with 6-carbon molecules. So 6-carbon carbohydrates. A kinase is an enzyme that transfers a phosphate. Okay, so here we have hexokinase. It's transferring a phosphate from a 6-carbon molecule to another 6-carbon molecule. So where's the phosphate coming from? It's coming from ATP. So here we're using an ATP. Okay, so we've used 1 ATP. We've invested 1 ATP here. So then a couple more steps happen. Now we have phosphofructokinase. Now I want to kind of point out this enzyme. This enzyme is a key enzyme in this particular pathway. It's one of those enzymes that's regulated by feedback regulation. So at this step, we can see that another ATP is invested. Okay, so ATP is hydrolyzed to ADP again, and then another phosphate is added onto this molecule right here. And again, look, phosphofructokinase. So phospho, we've got the phospho from the ATP. Then we have fructo, we have fructose, and then a kinase, we have the transfer of that phosphate to the fructose 6-phosphate in this case. All right, so we've got this energy investment phase. Now in the energy investment phase, we can see that we're using 2 ATP. So 2 ATP are invested, and then we end up with these molecules right here. So the key molecule here, these kind of alternate between glyceraldehyde 3-phosphate, or G3P for short, and dihydroxyacetone phosphate, or DHAP for short. Now, ultimately, glyceraldehyde 3-phosphate is the thing that enters in the next step. So this will kind of alternate between these two. However, G3P will be in the following step. It's important to note that, like I said, in the energy investment phase, the final product is G3P. But it's important to note that 2 G3Ps are going to be formed here. So we have 2 of these. Now, every step after that, there are 2 of each. So you can see the little box. Okay, so there's like 2 boxes, one on top of another. So it's important to know, again, 2 of each of these are made, including the products and the reactants within these pathways. So here we have G3P. Okay, now we have an oxidation reduction reaction. Look, triose phosphate dehydrogenase. Whenever you see a dehydrogenase, that means oxidation reduction. So the G3P is being oxidized while the NAD plus is becoming reduced. So G3P is going to donate electrons and protons to NAD plus to make NADH. And since there are 2 G3Ps, 2 NADHs will be reduced to 2 NADH. NAD plus is reduced to 2 NADHs. And then, of course, we form 2 of these molecules at the process. Now we've got the next step. This molecule is converted to this molecule by the removal of a phosphate. So you can see there's 2 phosphates here. There's only 1 here. Where does that phosphate go? It goes into remaking ATP. So now we have these 2 phosphates. Each phosphate is going, well, there's 2 molecules, right? So 1 phosphate, it will be removed and then combined with ADP to make ATP. So here, again, this is the energy payoff phase. We're making 2 ATP. So we've used 2 ATP. Now we've made 2 ATP. We've broken even now. Okay, so now we're even Steven. Now we wait for the next few steps, and then the final step, we make 2 more ATPs. So here we see that final phosphate is going to be removed from this molecule, phosphoenol pyruvate, or PEP, and then be transferred to ADP to make 2 ATP. So at this point, right here at step 7, we're broken even. Now at step 10, we have 2 extra ATPs. So we've actually made more ATP now. And now, once those phosphates are removed from those 2 PEPs, now we have 2 pyruvates. And this concludes the process of glycolysis. So this is that first. Okay, so remember, there's the energy investment phase. We invest 2 ATP. Okay, and then when we move into the energy payoff phase, we make 4 ATP. So we have a net amount of ATP of 2. During the energy payoff phase, we also produce 2 NADHs. These NADHs can be used to make more ATP later. So they're holding on to those electrons, and they're eventually going to transport these electrons to the electron transport chain. Okay, so these are electron carriers. And then again, the final products are pyruvate and water. It gives us an overview. The input for glycolysis is glucose. Okay, then we have that process. The outputs are the following. We end up with 2 pyruvate, we end up with 2 extra ATP, and we end up with 2 NADHs, which can potentially make another about 6 more ATP. All right. So that's really the summary there. The next step is pyruvate oxidation. So pyruvate oxidation is going to happen if oxygen is present. Okay. If oxygen is not present, something else happens to pyruvate. Okay. So we're going to talk about in the second segment, we're going to talk about if oxygen is present, we'll move to pyruvate oxidation, then the citric acid cycle. All right. So in summary, glycolysis, or kind of an overview of cellular respiration or aerobic respiration to be more specific. But we discussed glycolysis and the details of glycolysis. So remember, there are 2 phases. We have the energy investment phase and then energy payoff phase. Ultimately, the inputs of glycolysis include glucose, and then the output is 2 pyruvate. We have 2 ATP and 2 NADHs that are made. So the next couple segments will talk about what happens to those outputs after that.